Hi, I'm Nikolai Schwabe, CEO of ProImmune, and in this presentation I'll introduce to you our ProPresent assay for measuring antigen presentation by MHC to T cells. When we look at whether a protein sequence can cause a T cell response, then we have to remember that three factors are at play. One is antigen processing. Antigen presenting cells have to cleave proteins into peptide fragments and then load these fragments onto MHC molecules, a process which is limited by a number of restriction and selection elements, including a range of enzymatic and chaperone mechanisms. Two is MHC peptide binding. The peptides have to actually bind to MHC molecules in order for them to be ultimately presented. This will be limited by the MHC restriction of the MHC molecules present on the cell. And three is T cell function. In order to respond, functional T cells recognizing presented MHC peptide complexes have to be available. This is based on whether a T cell receptor with sufficient affinity is available in the peripheral repertoire, and any response is further subject to mechanisms of peripheral tolerance and other drivers of effector function. ProPresent is a flexible service that we have now been offering since 2011. This assay can directly measure the peptides that are presented on the surface of dendritic cells from a protein of interest. It's an in vitro assay that we carry out as a service at ProImmune, and we provide the donor samples for this. As such, it is the only commercial service that offers this type of analysis. We currently offer HLA class 2 analysis by subtype, also HLA class 1 and other species. One of the best things about ProPresent is that it is quick. For example, we can analyze 2 to 3 antigens on 10 donor samples in just 3 to 4 weeks, including providing a detailed analysis report. So how does it work? This picture shows an example workflow. Here we take a protein of interest and load it onto monocyte-derived dendritic cells. We generate these cells from our bank of HLA-type cryopreserved healthy donor samples. We then mature the cells to present peptides from the protein. Cells are then lysed and MHC molecules are recovered in an immunoaffinity step. We then release the peptides from the MHC molecules and sequence them in sequencing tandem mass spectrometry. The output is the peptide sequences, which often occur in nested sets as expected. So what can you do with ProPresent? First of all, it's a great way to demonstrate that suspected T-cell epitope sequences are actually presented on cells. It's also a tool for understanding immunogenicity more generally. You can look at how protein modifications modify antigen presentation, or the difference that allelic variants can make in proteins in relation to how their fragments are processed and presented. The effect of aggregation can also be studied. Although our standard method works with monocyte-derived dendritic cells, the method is flexible and we can really look at presentation from any cell type that express the MHC molecules of interest. In the context of immunogenicity, it is also possible to look at host cell protein presentation. To make sure that the assay works, we look for self-proteins that we know should usually be presented. For projects involving human disease, we look at several proteins that occur in most of the samples we measure. One of our acceptance criteria for the assay is that we find sufficient presentation from such proteins in each donor. To see how well ProPresent works in practice, we have used it to take a look at KLH. KLH is known to be a very strong T-cell antigen. It is around 3,000 amino acids in size. It's also a natural product that comes in two isoforms, KLH1 and KLH2. There's always been the presumption that it contains numerous T-cell epitopes, but due to its size perhaps, we did not know of anyone who has done mapping studies to identify these epitopes. So we put KLH onto 20 donors with our ProPresent assay, and then analyzed presentation by HLA-DR. Very quickly we found 20 new nested sets of sequences, of which some are shown in this slide. The table shows for each donor the HLA type associated with the donor. We also show the expect value for each of the sequences, which is similar to a p-value, with values lower than 0.05 indicating high confidence that the sequence has been identified correctly. The fact that you see such sequence nesting is also a good indication that the peptides identified are real hits. The sequences still have to be confirmed to be T-cell epitopes in T-cell assays, but at least we know that they can be seen by T cells, so they could well be relevant epitopes. We have also looked at therapeutic antibodies such as Humira. Humira is a fully human monoclonal antibody developed using phage display technologies. It blocks tumor necrosis factor alpha and is used for treating rheumatoid arthritis, amongst other indications. At $9 billion per year, it's also one of the biggest selling biologics worldwide. The labeling information for Humira suggests that it may generate up to 12% anti-drug antibodies in recipient patients. Recent research by a group of scientists at Sanquin in the Netherlands, referenced on this slide, shows that up to half of all patients on Humira develop neutralizing antibodies within around 6 months after starting the treatment. So the question is, why is Humira so immunogenic in practice? When we looked at Humira with ProPresent, we found that at least 4 peptide regions from the V region of Humira are presented on HLA-DR molecules. We have found 3 regions on the Humira heavy chain and 1 on the Humira light chain, and they are shown on the slide. This slide shows how the sequences we found overlay on the V region of Humira based on a homology model. As you can see, they reach into all parts of the variable domain, including the CDRs. It's perhaps not surprising that the CDRs of so-called fully human monoclonals contain T and in fact also B-cell epitopes, since those CDRs are not found as self-protein sequences in the recipient patient. From cartoon graphics of antibodies, it may also seem that the CDRs of antibodies are only a small portion of the overall molecule. But including flanking regions which can contribute to defining epitopes, they in fact account for around half or more of the total V region. CDRs are also designed to bind to something with high affinity, which is really the same as saying that something can bind to them with high affinity. And it seems logical that this something is not just going to be the cognate target of the antibody, but that it can also include anti-CDR antibodies that can develop in the recipient. Here we show an example of further analysis of one peptide from Humira that was identified with ProPresent, shown in green in the top table on the page. The top table also shows the HLA alleles that were present in the donors used in ProPresent that presented this peptide. We have followed up in more detail with both T-cell and HLA binding assays to characterize this peptide further. The bottom table with the traffic light colors shows the results of HLA binding assays we have carried out overlaid with the HLA types of significantly responding donors in T-cell assays. Red shows no binding, yellow intermediate, and green strong binding. After analysis with all 3 types of assays, it seems that this sequence is indeed a T-cell epitope, which is restricted for HLA-DR1 and DR3. So combining ProPresent with other orthogonal assays such as T-cell proliferation assays and HLA binding assays can really help to drill down to fully characterize new T-cell epitopes. Going back for a moment to a general level, I want to summarize the reason why ProPresent is such a great flexible technique. The reason is that it has 2 completely separable steps. Step 1 is the preparation of the cell sample that presents MHC molecules. At ProImmune, we have our standard method of generating monocyte-derived dendritic cells generated from our donor bank, which we can use, for example, to analyze sequences presented from therapeutic proteins. But alternatively, the customer can also produce their own cell samples from any species and by any means, so long as they are convinced that the method for inducing presentation works acceptably well, and so long as there is an appropriate immune affinity reagent available to isolate the MHC molecules of interest. In that case, all we need to do, the following step 2, is to receive a cryopreserved cell pellet from the customer. Step 2 then is a high-performance MHC peptide isolation and mass spec process, which essentially always works the same way. We recover the MHC molecules, sequence the peptides, and do the statistical analysis on the results. This separability makes the technique adaptable for many different applications. Here's a summary of the types of project that we have worked on so far. We have done a lot of work on a factor replacement protein. In this case, we have repeated the assay several times, and it was nice to see that the top scoring hits are essentially always the same. The results also compared well to what was seen in functional assays for the same peptides. We have also worked on heavily haptenated proteins. Here we were astonished to see how protein haptenation completely changed the antigen presentation spectrum compared to the unhaptenated control. Not only did we see modified peptides presented, but also many different unmodified peptides were presented compared to the normal control. It is clear that protein modifications not only affect the MHC binding of peptide sequences that comprise these modifications, but also that they can change protein processing, which can cause significant non-local effects in terms of which new and different sequences may now be able to be processed and bound as a consequence of a modification. Humira is just one of the recombinant antibodies that we've looked at, and in most cases we have seen several sequences that were presented. In another project, we have worked with a food allergen protein mixture. In that case, we were able to identify some key well-known allergen peptides from this allergenic food. And earlier this year, we have worked on a project where we have loaded DCs with a killed and also live pathogen. And again, we were able to identify a range of peptides from several different proteins. So in a sense, we're able to drill into the immunoepiproteome of a given pathogen to see how it can be recognized by the immune system. To conclude, by now I hope that I've convinced you that ProPresent is a great and fast way to find sequences of key relevance for the immunogenicity of a target protein, protein mixture or pathogen. The ProPresent technique can also answer questions that other assays simply cannot address in a clear and decisive manner. We think that it represents a key element in profiling any protein product candidate or therapeutic strategy that may affect T-cell responses as part of the development process. Typically, we turn around projects in only 3 to 4 weeks from the time we have received customer samples. And this unique service is only available from ProImmune. ProPresent is of course one of our cornerstone assays, but at the same time it seamlessly integrates with all the assays that we provide developed around the objective of being able to understand all aspects of immune responses. These assays include our ProImmune Reveal MHC peptide binding and T-cell assays, our ProArray Ultra microarrays for understanding and mapping B-cell responses, assays for looking at innate immune responses such as our cytokine storm assay, ProStorm, our pentamer technology that allows you to track single antigen-specific T-cells, as well as a range of supporting technologies including sourcing donor samples for projects, immune monitoring assays such as ELISpot and flow cytometry-based assays, HLA typing and peptide synthesis. And I invite you to visit our website to find out more about how our products and services can help complement your R&D efforts. Thank you for your attention, and we look forward to be able to help you.